So okay, so I think it's 9:30. So hello, everyone. Good morning, good afternoon, good evening from wherever you're listening from. Welcome to the Biologically Speaking webinar series. I'm Shaunak. I'm a postdoctoral fellow at the National Institute of Health, USA, and the co-founder of the Biologically Speaking series. And I'll be your moderator for today's session. Hello, everyone. Good morning, good afternoon, good evening from wherever you're listening from. Welcome to the Biologically Speaking webinar series. I'm Shaunak. I'm a postdoctoral fellow at the National Institute of Health, USA. Co-founder of the Biologically. Right, so those who are new to this forum, Biologically Speaking is an academic interest group initiated by a group of PhD students and postdocs from around the world, where we try to showcase amazing discoveries performed in the field of biological sciences and allied subjects, with an aim to initiate a scientific mindset among the students and also to promote scientific dialogue and collaboration between the researchers. All of our outreach activities are funded by the American Society for Cell Biology, and we organize scientific webinars, release blog posts, or cover a research article for general public. So if you'd like to contribute, please contact us through our website. We organize our scientific talks bi-weekly with a scientific theme every week. We had our inaugural session 2 weeks back with Dr. Nagraj Balasubramanian and Dr. Alexis Lomakin from the Children's Cancer Research Institute on matrix. And today we are carrying forward with the similar theme on matrix and moving on to morphogenesis. We are joined today by Dr. Abhijit Majumder and Dr. Akankshi Munjal. In September, we are joined with Professor Bruce Alberts for the cell in September 5. And for September 19, we have the sign-ups with Professor Upinder Singh Bala from National Center for Biological Sciences, Bangalore, and Dr. Poonam Thakur from IIT Trivandrum. And we hope we will join us for all of our talks. All of our talks are archived on our YouTube channel, so in case you were not able to join us via Zoom, please go to our channel anytime to listen to the talk. You can also visit our past webinars that we have hosted in this platform. Moving from matrix, our theme for today is morphogenesis, where we try to understand the physical force and role of underlying matrix on tissue development and physiology, and how cells respond to microenvironment and pattern during morphogenesis. Our speakers have kindly agreed to stay for a few minutes at the end of the session to interact with the audience. So this is your opportunity, and I'll strongly encourage everyone to avail that. A couple of housekeeping stuff before we start with the session. Please keep your audio muted and video turned off during the talk to preserve the bandwidth. And if you have any questions for the speakers, please write it them on the chat box and we will moderate them at the end of each talk. With that, it is my great pleasure to introduce our first speaker for today's session, Dr. Abhijit Majumder, Associate Professor at the Department of Chemical Engineering, IIT Bombay. Abhijit did his BE from NIT Durgapur in 2002 and an MTech in Cryogenic Engineering from IIT Kharagpur in 2004, following which he joined the lab of Professor Animangshu Ghatak and Professor Ashutosh Sharma to do his doctoral research on adhesion and soft mechanics, and earned his PhD from IIT Kanpur in 2010. He then joined Professor Josna Dhawan's lab in 2010 at INSTEM and CBS Bangalore for his postdoctoral fellowship, and also worked at PWH Harvard Medical School from 2012 to 2013 on cell mechanics and microfluidic devices. He has received numerous awards, including TIA 35 India Young Investigator Award and an Early Career Fellowship from the Wellcome Trust DBT India Alliance in 2013, and started his independent laboratory at IIT Bombay. Dr. Majumdar's lab's focus on how different mechanical aspects of a tissue control or influence the cellular behavior and cell fate, with an aim to understand the role of mechanics in pattern formation, development, and other pathological disorders. Abhijit is a member of the Global Young Academy and also has keen interest in science outreach, spreading scientific awareness among masses, particularly students. And today he will talk on cell migration and patterning in response to mechanical microenvironment. With that, thank you, Abhijit, for accepting our invitation to speak in this forum, and very much looking forward to it.
Thank you, Shaunak. Yeah, am I audible?
Yes.
Great. Thank you, Shonak, and thank you for every member of this biologically speaking group. And it is really a fantastic opportunity to have this more or less an informal, but as well as that discussion with all young minds here. So let me just share my screen, and you let me know if my screen is visible.
Yeah.
So is my screen visible?
Yes, perfect.
Yeah, great. So as Shanak said that we will be talking about the morphogenesis, so cell migration. So the theme of today's this meeting is morphogenesis. I will be talking about a particular work that has been done in our lab by my PhD student Akshada Khatpekar. He just graduated, few weeks ago. So now she is Dr. Akshada Khatpekar. So the whole work and like most of the slides that I will be showing here is directly taken from her defense. Before I start, this work is not in the true sense of morphogenesis, the way, in fact, Akankshi will be talking in great detail, after me. But what I will be showing here in this particular work is that how some very minute mechanical signals, mechanical microenvironment control both cell migration as well as patterning, and that I will show you the in vitro result. But we found this particular incident like quite fascinating, and that's why I would like to share that with you today. So form and functions, they always go hand in hand, right? I mean, everything in our body, they can do their the functions that they are supposed to do, whether it is skin, muscle, or even your blood vessels, anything, because they have a particular kind of arrangement, and the cells are arranged in a particular three-dimensional structure, or sometimes in a two-dimensional structures. But the structure is very important so that they can do what they are supposed to do. So how the pattern forms, that has always been an active area of research. And as most of you know, is that one of the major work was done by Alan Turing, and where he has shown that the reaction diffusion leading to formation of various kinds of cellular patterning, right? Now, when we are talking about this kind of cellular patterning, so there we talk about a chemical gradient, a chemical signal that is leading to cell patterning. And when we are talking about the cell patterning there, most of the time we talk about that cell type, that in one location I get one cell type, in another location I get another cell type. So this kind of this school of thought, here it is, the patterning is controlled by chemical signals, number one. Number two, it is the cell type dependent patterning. However, when we talk about the cell patterning, it is not just the various different cell types, but often the same cell type, they are physically arranged in such a way that they give rise to particular structure as well as functionality. The examples that I showed just in my last slide, for example, the muscle fibers, the cells are arranged in a particular way. That's why they form the fiber, and finally they can do those kind of muscle contraction, right? So, and so that is the physical patterning of the cell. And then this physical patterning or other kind of cellular patternings also often governed by the mechanical factors. By mechanical factors, I mean some kind of force, external force, internal force, force generated by the cells, how the surroundings are. Based on that, the patterning forms. One of that, example is the pattern on our fingertips, right? Our impressions, thumb impression, finger impressions. And these patterns are formed when we are in the mother's womb and the baby is trying to touch multiple, different surfaces. And when they touch multiple different surfaces, because of that, there is a force or some kind of buckling that happens because of this lateral force, which causes the buckling, and this buckling essentially we see as the pattern. So the pattern can be formed by the chemical signals, but it can also be formed by the mechanical signals. And I think again, Akankshi will be talking in great details from her very fascinating works. So I will show this video because I just love this video, and why I'm not being able to, just a second. So I'll just show this particular video, and yeah. So here you will see a Drosophila embryo that is kind of developing. I mean, this is one particular video that I really like, and I often show it even in my cell mechanics, the course that I take. So what to observe here is that the cells are dividing constantly. They are increasing in number, but also after a certain point, you will see a huge migration that is happening, right? A huge level of migration that is happening. And the pattern segmentation, etc., that follows after this, that is the result of that wide global scale migration. So we can understand that the formation of pattern or the structures during the development is the migration plays a very important role there, right? So with that, I will also would like to show you two other examples. This is from a much earlier paper, 2000 paper. This was, I think it is not PNAs, I think it was Biophysical Journals, my mistake. So here what they did, they took a material. So it is a two-dimensional material. So there is a cell and there is a cell, and they have created the soft and the rigid area. And here is a kind of a border or boundary, no physical, I mean, no boundary which is, there is any topology in that boundary. It is absolutely smooth. However, if you touch this side, it is soft. If you touch this side, it is rigid. Same here. And then they observed how the cells migrate. And what you can see here, when the cells are trying to go from soft to the rigid zone, they can easily cross the boundary and they can move from soft to the rigid zone without any problem. On the other hand, when the cells try to go from the rigid to the soft, they actually cannot go. They touch the other side and then they turn and comes back. So this, they have termed this phenomena as durotaxis. Duro meaning the rigidity and taxis, obviously the movement. So since then, from last 20 years, many people, they have shown the effect of substrate rigidity on various different cellular behavior, including cell migration. However, here we ask a little different question. Here we ask that instead of having a purely soft or purely rigid kind of structure, what if the structure is inhomogeneous? What do I mean by inhomogeneous? Here you can see that if I have a soft gel, what I have here is polyacrylamide gel, the stiffness of which you can control by changing its cross-linking density. But in that soft gel, I embed a glass bead, which is orders of magnitude stiffer than the gel. So this is how it looks like. Now, on this gel, I have to put a ECM coating, otherwise cells cannot adhere to this gel, but then I put the cells and they do adhere to this gel. So I am asking the question here, what will be the effect of such kind of embedded rigid structure in an otherwise soft microenvironment? Okay, why we are interested in asking this question? Obviously just for fun, because many of the times we do things just out of our curiosity, but also if we look in our body, any place for that matter, it is not a single stiffness that is in that region. It is always a combination of stiffness, right? It is, and many of the time, the rigid or soft, the part is embedded inside the other microenvironment, right? Which you can also see here, say in case of a cancer. Generally, cancer tumors are much stiffer. And that is how in many of the solid tumors, that is first kind of the doctors realize that there is a tumor by simple touching and pressing it, that okay, there is some kind of lump, right? So tumor is also such a situation where it is an embedded structure inside a soft environment. So that is what we would like to ask the question here. So what do you see when we put the cells on this? So if the cells, so here I have C2C12 cells, it is a mouse myoblast. Here on plastic, they are aligned, but there is no global alignment. Similarly, when I put it on a soft gel of but uniform stiffness, again the same thing. There are some kind of local alignment, but there is no, global structure. But as soon as I have a rigid material at the center, somewhere here, this is what we get. That this black thing is an embedded bit. It is not close to the surface or even at the surface. It is 200 micron below the surface. But still, cells which are at the surface, they can not only sense that, okay, something is there, they actually arrange, they align, and then they finally arrange themselves in these beautiful sunflower-like structures, right? So, and so if we put the bead, we get this sunflower kind of structure, but we can control, and that is very interesting, that we can control how this arrangement will be just by controlling the what I am embedding inside. If I put a glass capillary, cells on the capillary gets aligned axially, and then side they get aligned like a feather. Radially, you can have various different combinations, and you can get various different kind of cellular alignment. So, and what was very fascinating for us, that this long range of patterning. So this is the same image, I'm just showing it in a colorful way, just to that what you can see here, that all the when there is no inhomogeneity, whether the material is very rigid or soft, we only see the local pattern. However, when there is a some beat at the center, we see a very long range patterning. And how long it is, we can clearly see that they are 1,000 or even sometimes 2,000 micron long, depending on what radius of beat I am putting there. And if I think of the length of a cell is 20 to 30 micron, we are having some 35 to 40 cell length long patterning. So it is really a long range patterning that we are seeing here. So this is the same result. We have just did some kind of correlation length to see that for at what distance the cells are correlated. So that is what we see here. So then we would like to see that, so all of this pattern formation that you saw, this is the end product, meaning is that after putting the cells on our substrate, so this image you are seeing what happens after 48 hours, when it is the cell have formed a complete monolayer. But then we wanted to ask the question, what happened in between, how it forms. And this is the video that you can see how it forms, that 48 hours video. So this is how the patterning is forming. However, instead of a soft gel, which is 2 kilopascal, if I take a much rigid gel 10 times stiffer, 20 kilopascal, this is what we see. We don't see this kind of nice pattern formation that we see here. Okay, it is again, so the cells actually cannot sense that there is a bead inside. So we further wanted to know what is happening. So this time we put a much less number of cells, and this is what we see that things are happening. You can clearly see that cells are actually migrating towards they are aligning, but not just that, they are also migrating towards the beat. And that is what we are further interested to know, that what is happening, how they are migrating towards the beat. And here what we just measured, that the cumulative displacement, and if it is the negative, then they are moving towards the beat. If the displacement is positive, they are moving away from the beat. And as you can see that the cells, and then the whole location that we have divided into multiple zones, right? And each zone is of each annular zone is of 200 micron width. And you can see here that for first 3 zones, cells are nicely migrating towards the beat. And for the cells which are further away, they are not sensing the beat, as we kind of also expected. But we can see here that there is a directed biased migration that is being caused by this beat, and that effect is actually quite far away, working even at 600, 700 micron away. So we wanted to know that what is happening. So we checked for various different parameters. In the interest of time, I am not showing you all the result, but I will quickly kind of take you through that. We have seen if I increase the rigidity of my material gel, as I have also shown, this biased migration and the pattern formation go away. Also, if I make the material viscoelastic, that is, when the cells, they apply force on the material, they deform the material. But if the material is viscoelastic, that stress will get dissipated, go away, and then also they cannot form the pattern. Similarly, bead size, shape, substrate thickness, all these are making some effect. That tells us that it is a geometry and the mechanics that is playing an important role. We saw that protein coating on the gel does not make any difference. Whether it is collagen, fibronectin, laminin, every time the cells get nicely aligned. Also, another thing that we have noticed, that this bead and the gel that we have, if we coat my bead with a hydrophobic material so that the bead and the gel is not tightly adhered, then such pattern does not form. Also, we understood that the cell shape and cellular tractions are important. So we would like to know that if this pattern formation is because of some kind of ECM gradient or rigidity gradient. We found no, it is not. So we started looking further, and then we realized that because of the bead, what is happening is that in this region, because this gel swells, we are actually getting a differential swelling. On top of the bead, swelling is less, but further away from the bead, where the material is free to deform, there they swell. And because of the swelling, a strain gradient that gets generated. So we thought that yes, the strain gradient is playing a role. But if the strain gradient was the only parameter, then the cell shape and cellular traction, those should not have played a role. So then we kind of realized that yes, the strain is important, but cells are also important. And this is something that we already knew from our own work and from others' work, that yes, cells can sense the strain. And they also can form different kind of structures by sensing strain caused by each other. So what we did a very simple experiment. We just changed the number. So far we were having 3000 cells per centimeter square. Now we have only 500 cells per centimeter square. And we see that as we decrease the number, the biasness in the migration goes away. So now the 50% of the cells are moving towards the beat, 50% of the cells are moving away from the beat. So there is no bias, but you can see the bias only when you have some critical number of cells. And we have also seen that if you increase the cell number too high, then they start to kind of collide with each other and the biasness go away. So this is, so with this understanding, what we propose here is that when we have a bead, because of this differential swelling, you get some kind of strain gradient, or the mechanical stress that gets generated in the microenvironment. When, because of the strain, and also it is not an uniform strain, there is a strain gradient. And because of the strain gradient, a circular cell senses that strain gradient and it becomes elongated. However, just only one cell getting elongated is not enough. You need enough number of cells together, because when they get elongated, they also apply force on their material. So they deform the material further. And then they apply the strongest force at their two end. So as a result, they essentially reinforce the stress gradient or strain gradient that is there inside the gel. So they reinforce the strain gradient together. And as a result, they align one after the other. So it is a kind of a feedback mechanism that helps them to align, or you can think of that it is some kind of crystallization that happens, that the things are, in a saturated, you first of all, to get a crystal, you need a saturated solution, but just having a saturated solution might not give you a nice crystal. You need to give something to nucleate. And as soon as you give a point to nucleate, from there, things can spread around. So that is what we see here, and that we have put in the archive. So there are a lot of details there. So if anyone is interested, they can go and have a look there. So with this, I stop here. So what we have seen is that the mechanics and cells, these are two interacting worlds. And as a result, they do a lot of the stuff, but including what we have shown, that the migration and the pattern formation, that is a very important starting point for morphogenesis. So this is my group, and I thank all my funding agencies, my group, and particularly Akshada, who has done all the hard work that I have shown you today. Thank you.
Yeah, thank you, Abhijit, for that interesting talk, understanding how the underlying matrix and the substrate rigidity can affect the pattern formation. It's really interesting. If anyone has any questions, please write it on the chat box and we can discuss with Abhijit at the end of the session. But I can have one question because we have some time for this part. Okay, Shaunchari has a question, so maybe I can just ask that. I'll keep mine for the audience interaction. So Shaunchari is asking, the small lump or tumor leading to formation a big one. I'm actually not sure what that means.
No, probably she is kind of suggesting that if that is possible. So I don't have a evidence for that, but what we kind of think of, that it might be possible that because a stiff, the tumor is somewhere, maybe the angiogenesis, which we know that from the nearby blood vessel cells migrate towards the tumor due to the VEGF signaling, but maybe the mechanics might be playing some role there. So it is quite possible, but I don't have an evidence for that.
Right. If anyone else has any question, then what are the genes responsible for the patterning from Muskani Tahira?
We have not looked at all the, the gene structure, or in fact, I'm a chemical engineer and I work on the cell mechanics aspect. I'm more interested to understand how the mechanics, the mechanical factors. So yes, it would be good to look at the gene, but yeah, we are not looking into that.
Right. Okay, so I think we can move on to the next part, Abhijit. I hope you will join us for the audience interaction, and then we can have more discussion with the audience. Thanks again, Abhijit, for that exciting talk. Moving ahead, we have our next speaker for morphogenesis, Dr. Akankshi Munjal from Duke University School of Medicine in the United States. Akankshi did his undergraduate from University of Delhi and the master's from NCBS Bangalore from the lab of Professor Satyajit Mayer in 2011. And thereafter, she joined the lab of Professor Thomas Lecuey at Aix-Marseille University in France, where they showed how actomycin networks lead to changes in cell shed during tissue morphogenesis. Akankshi earned a PhD in 2015 and joined Professor Sean Megason's lab at the Department of Systems Biology at Harvard Medical School with the HFSP long-term postdoctoral fellowship. She utilized zebrafish inner ear model to identify the local secretion of the extracellular matrix enzymes can drive tissue morphogenesis. In 2019, she received the K99R00 grant from the NIH to establish her independent laboratory. And early next year, she will be starting at the Duke University School of Medicine, and her lab will be utilizing quantitative imaging, theoretical modeling, and sequencing approaches to uncover principles of morphogenesis. So anyone looking for a position or want to collaborate, this is your chance to interact during the audience interaction session. And Akankshi is actively involved in science outreach and is also co-founder of the CISBio postdoc group, discussing different opportunities and career development of postdocs. With that, today she will talk on uncovering principles of tissue morphogenesis with the zebrafish inner ear. And without further ado, let's hear it from Dr. Akankshi Munjal. Over to you, Akankshi.
Thank you. I'll just start sharing my screen quickly.
Yeah, it's
all right.
visible.
Do you?
Yeah,
awesome.
Perfect.
Yeah. Let's start the timer again so I don't go over. Okay, thank you so much, Shona, for the introduction. And I want to congratulate the biologically speaking community for this commendable effort and the organizers for this wonderful opportunity. I'm really looking forward to the discussion. So please feel free to ask all kinds of questions. There's no such thing as naive or dumb question. Okay, so I'm interested in understanding how complex three-dimensional organs with very robust forms form from simple embryonic tissues. And since the form of an organ is tightly related to its function, understanding the design principles of organogenesis is important not only for healthy development and physiology standpoint, but also to improve our understanding of developmental disorders and pathologies. Over the years, different research communities have addressed this question to build a framework, which I'll break down into three important parts. And Abhijit has kindly introduced several aspects of this framework, so it makes my job easier. The first part of this framework is that involves often identification of developmental signals, which are either morphogens or transcription factors. And these signals then pattern embryonic tissues to confer unique properties, such as polarized distribution of intracellular forces and or cell adhesion. And this pattern cell mechanics would then drive cell behaviors such as cell apical constriction or cell intercalation, which then result in events such as bending or elongation of tissues. And again, Abhijit showed a fantastic video of the early development of Drosophila embryo, and I'll try to use this framework to say that it really epitomizes what this framework is talking about. So in the early Drosophila embryo, the expression of transcription factors, snail and twist is required for the ventral patterning of the embryo. And these transcription factors have downstream targets like the ligand FOG, which binds with GPCR such as smog and mist. And during my PhD, in collaboration with Stephen Kerridge in the lab of Thomas Lecuey, we showed that this GPCR signaling is responsible for medial apical accumulation of intracellular forces that drive apical constriction and ultimately lead to the invagination of this ventral tissue that then goes on to become the muscle layers. So this framework has been extremely useful in understanding morphogenesis over the last few decades. But more recently, several drawbacks have emerged. The first limitation of this genetic information-centric framework is that it fails to account for emerging behaviors. For example, accumulation of intracellular forces in the form of actomycin contractility is often associated with emergent behaviors like pulses, waves, and flows. And these dynamics have been observed in a variety of developmental contexts across species. And during my PhD, I found these dynamics to be self-organized in that they arise from local mechanochemical interactions between myosin and its regulators rather than an upstream genetic pacemaker. Another beautiful example of emerging behavior is when cells segregate with cells that have similar adhesion in order to minimize surface tension. And this differential adhesion-mediated cell sorting allows tissues like the neural tube to form sharp boundaries, even if the developmental signal, in this case, sonic hedgehog, has a noisy pattern. The second drawback of this framework is that it puts intracellular forces generated by actomyosin networks at the center for force production, and it fails to account for extracellular forces. And more recently, several groups have shown that hydrostatic pressure created by water in luminized tissue can also provide forces necessary for morphogenesis. For example, hydrostatic pressure can power expansion of the mouse blastocyst during its early development. And in my postdoc work, I will show you the role of the extracellular matrix in providing forces necessary for tissue morphogenesis. Finally, the third major drawback of this framework is its hierarchical nature. The pathway, as I show here, is not really a pathway, but a dynamic network where, due to morphogenesis, cells are moving, causing the pattern to change, and these forces may be transduced to change the developmental signal. And these feedback interactions can play important roles in fate determination and robust shape formation. So in the next 15 minutes or so, I will try to convince you that the semicircular canals of the inner ear is a fantastic model organ to address these gaps. So the semicircular canals of the inner ear are required for sensing balance and acceleration. And this function, as you see in these movies, depends on its precise structure. And defects, malformation, and infection in canals can cause vertigo and dizziness. And semicircular canals can also develop abnormally in association with several human syndromes. Yet, despite these associations, the cause and the etiology of these abnormalities have remained unknown. And how healthy development of inner ear or semicircular canal happens is also unknown due to inaccessibility of the inner ear in most model organisms. So all vertebrates have two sets of canals in each ear, with their numbers ranging from 1 to 3 per ear. All jawed vertebrates like us have 3 mutually orthogonal canals in each ear. And their structure is quite conserved, as you see here in these pain-filled stains. I study them using zebrafish as a model. And before I tell you why zebrafish is an ideal model system to study inner ear development, I just want to take a slide to show you why zebrafish is an excellent quantitative model system in general. So this tropical fish found in Ganges can lay hundreds of eggs that are externally fertilized, convenient for genetics, and are transparent, making them ideal for light microscopy. Their development is quite rapid. So within a day or 24 hours, you'll see that this single cell embryo can form a fish-looking larvae. And besides making beautiful movies like these, it's possible to do quantitative measurements. So we can segment and track individual cells, measure protein and gene dynamics in those cells. It's also possible to apply and measure forces, for example, using laser ablation. We can also measure material properties by injecting these oil microdroplets. And more recently, it's possible to map the transcriptome of the entire developing embryo using single-cell RNA sequencing. So the reason why we think fish is an ideal model system to study inner ear development is because fish have inner ears. But unlike most vertebrates that have middle and outer ears, fish lack middle and outer ears, making their embryonic inner ear both optically and physically accessible. So most of what we know about semicircular canal development comes from Tanya Whitfield's group using genetic samples and using genetic mutants and fixed samples. But with light microscopy, we can study the morphogenesis live at high spatial and temporal resolution. So the life of the inner ear starts off as a thickening of the ectoderm, which corresponds to the embryonic otic placode. And in this movie made by my colleague, you will see that this placode undergoes cavitation due to influx of ions and water that build hydrostatic pressure. So this is another example of how hydraulics is controlling the size and shape of the tissue. So this tissue will continue to expand due to pressure and cell division to form a football-shaped structure called the aortic epithelium, so an American football-shaped structure. So this is a single-layered tight epithelial structure. This simple tissue will now undergo a massive topological transition to form the semicircular canals. And I'm interested in understanding how this transition happens. So we will look at this transition live together. Here I'm using a membrane-labeled transgenic fish, which is mounted dorsolaterally such that the objective is coming from the top. I acquire Z sections, render them 3D using this open source software by flow render, and only look at the top or the dorsal half of the epithelium where morphogenesis is happening. So this is the aortic epithelium. It's sandwiched between the hindbrain and the skin and has this invisible lumen. So morphogenesis begins when six regions in the aortic epithelium begin to sequentially bud into the lumen. So first these lateral buds form, then the anterior and the posterior bud form, and later on you'll see that this ventral and ventrolateral buds are forming. So let this movie loop again. These buds anisotropically extend towards each other. So this lateral bud will meet this anterior bud, this posterolateral bud will meet the posterior bud. They then fuse to each other, as you see here. So from a single sheet after the fusion, you will see that there are 3 points of contact. So they form these pillar-shaped structures after fusion, and the negative space around them corresponds to the semicircular canals. So I just drew these cartoons to help you imagine the topology. Here is the aortic epithelium showing 2 representative buds. The buds extend, fuse to form pillars, and the space around them, this lumen space, lumen-filled space, corresponds to the canal. So when we move, this mechanical signal is transduced by hair cells into electrical signal, and this is how we sense motion. So what is the mechanics of this process? This is a multi-scale problem, and just so the topology is clear, I will go back and forth between these 3D rendered images that I just showed you and 2D sections where the lateral buds are in the more dorsal planes or the top planes, and the anterior and the posterior buds are in the more ventral planes. So to save time, I will tell you that we systematically ruled out conventional actomyosin-driven modes of morphogenesis, such as proliferation and cell intercalation, and found that morphogenesis of each of those buds that I just showed you is locally initiated, such that if I use a laser to ablate a single or a few cells in a nascent bud, in this case, I'm ablating a few cells in the anterior bud, it's sufficient to block the extension of that bud, whereas the adjacent non-ablated buds continue to form and extend. So what is the force that is driving morphogenesis of each of these buds locally? Almost 30 years ago, a beautiful study by Haddon and Lewis showed the role of hyaluronic acid in frog inner ear morphogenesis. But the genetics and the mechanics of this process has still remained to be understood. So hyaluronin is a glycosaminoglycan. It's a major constituent of the extracellular matrix. Its substrate are synthesized in the cytosol with these enzymes UGDH and UGT. And these substrates, which are essentially sugars, are polymerized by synthases that sit at the plasma membrane, which extrude out large chains into the extracellular space. So this is different from how proteinaceous components of the extracellular matrix are made, and is similar to how plants make cell wall using cellulose synthases. So I checked the localization of these enzymes UGDH and HAS and found that consistent with our local initiation model, UGDH and HAS3 are present exclusively at the sites of morphogenesis. So here you see them in green and magenta at the sites of budding, whereas collagen 2, which was my positive control, is expressed by all the cells of the aortic epithelium. So here I want to give a shout out to this wonderful technique developed by Choi Group in Caltech, where you can use HCR probes for fluorescent in situ hybridization, allowing to see up to 4 or 5 genes in a quantitative manner in a developing embryo. Okay, so the next step was to check loss of function of these enzymes. And indeed, using either a morpholino-mediated knockdown or a mutant, I find that both UGDH and HAS are essential for semicircular canal morphogenesis. And I've confirmed these with CRISPR-Cas9 against HAS3 as well. So how is hyaluronin, which is, as I said, essentially a sugar, driving morphogenesis? So hyaluronin is a really interesting molecule. It's an anionic biopolymer that attracts sodium ions and water through osmosis to form a swollen hydrogel that applies pressure on the surrounding tissues. And it's for this reason it's used a lot in skin treatments. The hydrogel has unique properties, which means that unlike collagen or fibronectin, which are mostly stiff or elastic elements of the extracellular matrix, hyaluronan is not only stiff, but also viscous, which means it can deform. So these viscoelastic properties make hyaluronan an excellent candidate for tissue remodeling. So using a binding protein that has high affinity for this sugar, remember this is not an antigen antibody reaction, I find that hyaluronan is expressed exclusively in this extracellular matrix just beneath the cells that are budding. So here you see it in purple. Whereas collagen 2, which was my positive control, is expressed in the basement membrane and the extracellular matrix surrounding the entire auric epithelium. So how is hyaluronan driving morphogenesis? So what's happening in controls is that as cells are depositing hyaluronan, the buds extend and the bud ECM volume increases, as you see in this movie here. And I can digest this matrix using hyaluronidase that breaks down the polymers into oligomers, and I find that this causes the bud ECM, which is this hyaluronan-rich ECM, to collapse, and the buds in this case now fail to extend. Another observation we made was that as buds are extending, the cells stretch in longitudinal and circumferential axes and thin in this radial axis. And this stretch or strain rate that the cells are experiencing is hyaluronan dependent, such that when I digest hyaluronan, like in this case, the cells lose their stretching in the circumferential and radial axes. So with these and other experiments, we concluded that hyaluronan is driving morphogenesis because of its osmotic swelling. It's absorbing water due to its negative charges that cause dispersion. Another observation we made was that as buds extend, they extend about 3 times in their longitudinal axes compared to their circumferential axes, which remains roughly constant. So we asked, where is this anisotropy coming from? To save time, I will tell you that we ruled out a hyaluronan-based anisotropic extension model, primarily because hyaluronan is driving morphogenesis through pressure, which by Pascal's law is isotropic. Moreover, the genes that synthesize hyaluronan are expressed uniformly by all the cells that are budding. Instead, we turn towards looking at the epithelium and ask whether these cells that are budding can cause anisotropy. So epithelial cells can produce, sense, and respond to forces through their cell cortex, which is composed of actin and non-muscle myosin motor proteins. Using live transgenic reporters, I find that both actin and myosin are normally present on the apical surface, which is the surface facing the lumen of the auric epithelium. But during morphogenesis, both actin and myosin accumulate basally, which is the surface facing the hyaluronan-rich bud ECM. And to better resolve this basal accumulation, I did mosaic labeling. And to my surprise, I found a lot of actomyosin-rich protrusive activity. These protrusions, as you see, are not going through the extracellular matrix, but rather extending beneath the basal surface of adjacent cell. Remember, this is a non-migratory epithelium, so we were not expecting to see these dynamic protrusions. And indeed, when we search literature, we find examples where protrusions are present in non-migratory epithelia, for example, in cytonemes, and more recently, they've been shown in closing of the heart by the opposing tubes to match cells from Timothy Saunders' group. So in our case, we see that some of these protrusions, as you see here, are making contact or tethers with protrusions from neighboring cells. And these tethers, as you see, are naturally retracting. We find that this natural retraction of these tethers is in fact hyaluronan dependent, such that as the matrix is expanding, it causes these tethers to break. And when I remove hyaluronan using hyaluronidase, these tethers fail to retract. So instead of shooting these tethers with lasers to ask if they're tensed or not, we use their natural retraction in response to hyaluronan and ask if they have tension. And indeed, we see signatures of tension, where as tethers retract, their adjacent membrane angle widen, and they have a recoil distance between adjacent cells, which are both signatures of tension. So we named these tethered protrusion cytocinches to contrast their mechanical function, which is to hold tension, from the signaling function of cytoneins, which are involved in chemical signaling. Okay, so are cytocinches responsible for an isotropic bud extension? They can be if they have higher distribution in the circumferential axis, constraining pressure from hyaluronin in this axis and allowing extension in the orthogonal axis. So indeed, when I measure individual cytocinches during the extension of the bud, I find that both their numbers increase and their angle become progressively biased in the circumferential axis, such that when I do a product of these two, it correlates very, very well with the anisotropic aspect ratio of the bud. And is this sufficient to explain an isotropic bud extension? For this, I collaborated with an awesome physicist, Edouard Hanesso in IST Austria, who built a 2D vortex model simulating the lateral apical and basal tension in a slice of epithelial monolayer. And indeed, in this monolayer, when he adds this inward tension coming from cytocinches, which is the only free parameter of the model, this is sufficient to drive the extension of an isotropically shaped structure into an anisotropic bud. And indeed, when he simulates parameters and estimated values of tension from experimental data, he finds that it correlates very well with the experimental data. So prediction from the model was that if you remove this inward tension, the structure should relax to a hemispherical shape. And we tested this prediction using cytochalasin D, which depolymerizes surfactant, and find that using a mild inhibition, I was able to inhibit just the cytocinches, but still maintain the epithelial integrity. And in this condition, you can see that the bud now becomes more hemispherical, validating the prediction from the model. So what I've told you so far, just to summarize, is that certain cells in this auric epithelium are producing the unique hyaluronin-rich matrix. I still don't know the identity of this upstream genetic pattern that causes these cells to express hyaluronin. This hyaluronin then creates isotropic pressure, causing the overlying epithelium to form these buds. And the epithelium in return produces these tethers or cytocinches that are actomyosin-rich in the circumferential axis, constraining pressure in this axis and allowing extension in the orthogonal axis. And I have more recent data showing that there is ecadherin clusters localized in these tethers that causes them to remain attached to each other. And just to put this perspective in the broader, context, the field of morphogenesis had been dominated with studies which show how developmental signals directly pattern contractility and adhesion to drive morphogenesis. But more recently, with the role of extracellular matrix, hydrostatic pressure, and tissue-tissue interactions, a new theme is emerging where any or a combination of these components can create uniform force. And the interplay of this force with pattern stiffness, again, not only coming from ECM, but also from neighboring tissues, or in my case, I showed you actomyosin networks, it can result in a wide variety of shapes that we see during morphogenesis. So with that, I'd like to thank my mentors and collaborators, extremely generous sources of funding. And I've been very lucky to have worked in extremely stimulating lab environments at Harvard in France, and as well as in NCBS in India. And as Shonak already mentioned, I will be starting my lab at the Department of Cell Biology in Duke University School of Medicine, and we have positions available at all levels. So just check this website out, and if you're interested in understanding morphogenesis using zebrafish as a model system, please feel free to write to me. And with that, I'm happy to take questions.
Right, thank you, Akankshi, for that exciting talk. I truly enjoyed it. Discussing the feedback control between developmental signal patterning and morphogenesis. Such fascinating videos on aortic epithelial morphogenesis. We have got a lot of questions, so we can start. Okay. But I have a very naive question to ask: is the So this bud formation stage that you use for the semicircular morphogenesis, it has a lot of similarity with other ectodermal appendages development. So do you think it's all about the mechanical forces that act differently, leading to the development of different structures, or it is purely driven by the growth factors and the morphogens?
Yeah, that's like, what is the pattern here is an important question. So there are, like, as you said, in biology, we see a lot of tubes and we see a lot of buds. So lungs, when they are undergoing morphogenesis, branching morphogenesis, they produce these buds, which are the points where the future branch will grow to form. And there, it's known that it's a combination of growth factors as well as spatial constraint from the surrounding mesenchyme. In the case of the aortic epithelium, in fact, the aortic epithelium is also surrounded by these mesenchymal cells. We don't know what there are, what their origin is. My feeling right now is that it's likely to be a combination of development and mechanics, which is the case in pretty much all developmental questions. Another thing that I'd like to add here, which I didn't mention in the talk, is that we've done single cell RNA sequencing of the developing inner ear at 4 different stages. And with that, we have some likely candidates of what the transcription factors or growth factors might be, whether they're secreted by the aortic epithelium or the surrounding mesenchyme that could pattern which sides of the aortic epithelium will form these buds. So this is something that I will pursue in my own lab. So if anyone is interested in testing out these candidate genes, I'm happy to discuss more.
Great. I think with that, we can also invite Abhijit to share this podium, and we can have the audience interaction together. So we've got a couple of questions. So I can ask you to unmute, and if you want to ask a question by yourself, or if you want me to ask, I can also do that. So just let us know. So I'll start with Abhijit questions itself for the audience interaction. So Abhijit, you can only ask that to Akankshi.
So Akankshi, so like, fantastic and the fascinating talk, and it was very exciting to see. So I mean, my question was that you digested the I can't pronounce those names.
Hyaluronan, yeah, you can call it HA. Yeah.
HA, yeah. So is it possible to do some genetic mutation that you don't digest, but they just produce less or produce more?
Yeah, I've tried that a lot, not genetically, but with chemical perturbation. So I try to precipitate out hyaluronan using polycations so that they balance out the negative charges. None of those experiments have worked. It's just fish is a very robust, robust little embryo which can equilibrate pretty quickly whatever you inject. Genetically is a good idea. So the enzyme that is responsible for HA production in the ear is Has3, which is known to form intermediate lengths of the polymer. So just intermediate length for a HA is still 1 or 2 megadaltons, which is huge. So I think like the experimental thought you're going towards is expressing maybe another enzyme like Has1, which is known to make shorter lengths. And it's true that the hydrogel properties that are directly affected by the molecular weight of HA. So that could be an interesting way to modulate the physical properties, and we haven't tried that, but it's worth pursuing. Yeah.
Thanks. Thank you.
Thank you for the question. Yeah.
I'll move on to the next question for Abhijit. Arnav Saha has a question. So Arnav, I've asked you to unmute so you can ask the question directly. Arnav, can you hear us?
Arnav said that his mic is not working.
Oh, okay. So I can ask the question. So a curiosity-driven question to Dr. Abhijit in context of the role of matrix stiffness in homogeneity and your suggested model on SM-ataxis. Can we get some insights into why do specific tumor types, such as HA, behave differently from other tumor types? And characteristically metastasize to distinct tissues, and where the tumor cells mechanically predetermine their metastatic sites.
Yeah. So I think Arnav, so that's the answer that Akankshi gave. So I will kind of copy that answer once again. That in most of the cases, it appears and it looks like that it is a combination of chemical, biochemical, and the mechanics, right? So it is already known that the tumor metastasis and how the tumor goes from one, so that they are going out as well as invading, both are controlled by the tissue stiffness. Where it will metastasize, whether the mechanics plays a role or not, I think that is an open question. But at the same time, it is known that the homing is mechanics dependent. So, I will not be surprised that if it is at least to some extent controlled by the mechanics. Akankshi, do you have anything to add here?
No, I think Abhijit, it's there. I mean, there are now more and more genes coming up. GAP has been very popular, but even ERK signaling and in my single cell data set, we started to find more genes that are responsive to mechanical input. So it's very realistic now to imagine how chemical signaling can receive mechanical input to change downstream expression.
And also in one of your slide, as you are talking about the feedback, it looks like there is a lot of the feedback things in there.
Yes. Yeah. That's something that that I would really like to pursue in my lab, because we have a candidate gene that could respond to stretching of these cells as they are forming, but we don't know what the downstream effect of that would be. So we see expression of this gene in the buds, but we don't know what are the downstream cell behaviors that it's causing. Yeah.
Okay. I'll ask next Neha Vyas for question for Akankshi. Yeah, I've asked you to unmute if you want to.
Hello, can you hear me?
Yeah.
Hi, Neha.
Hi, Akankshi. Very interesting work. Enjoyed every bit. What I'm curious about is what leads to a very specific expression of HA in the bud?
Yeah, that is the question. This is why we ended up doing the single cell RNA sequencing. Unfortunately, it's not a simple answer. Like, if I could just say like, oh, it's I don't know, BMP4 causing HA expression there. It's unfortunately not a straightforward answer. It's likely an intersection of a bunch of morphogens. Because there is hedgehog coming from the hindbrain, the mesenchyme is likely producing FGF. Otic epithelium itself produces FGF. So yeah, we don't know the answer. But what's interesting is that it could be like, again, a combination of just like signaling to say that, oh, these are the specific bud sites, but we see hints of a Turing-like pattern because the spaces between the buds are quite uniform, and there are landmarks with respect to how far or close they are from the hair cells that are responsible for transducing this lumen movement when we move. So yeah, no, it's going to be a very messy answer, unfortunately, but that's what biology is.
Yeah. Thank you so much.
No, thanks, Neha, for the question. I have a question for Abhijit. It's like, you have shown that the cells actually elongate with when the bead is nearby, and then it kind of makes this pattern. But yeah, as you mentioned, that it also leads to pressure to the cell as well as it elongates. And there are a couple of reports lately that this pressure can lead to some sort of DNA damage and genome instability through the nucleus. So do you actually think in order to reduce the DNA damage and getting apoptotic, the cells kind of have a mechanism to align it together and then pattern?
So number one is that here the pressure or the stretch is not external, right? It is not an externally driven stretch. So where the DNA damage can happen, or like, you know, all those experiments that you are talking about. Here the cells actually have an option to kind of change and, you know, change its orientation. Being said that, as you said, that it is energetically also favorable for the cells to align its principal axis with the strain axis. And if it can align with that one, then also the nucleus get aligned. And as you said, then it is a bit, I mean, both from energetics point of view, as well as damage point of view, it is better to kind of preserve that, right? If
Right.
it can align in that way. So that is how a single cell that aligns itself. And when a single cell align itself, as I was talking about, that if they are at the multiple, I mean a second cell here, then again, if it does not align itself, then there is a mismatch. And again, the same thing kind of goes on. And that is probably leading to a long range of, and what I have not shown here in my presentation, that there is also some kind of relay is involved, that the first layer of cells, they get aligned, then the next and the next. I have not shown that because the data is not very clean. So I have my own doubt there, but we are trying to kind of understand that. Yeah.
No, that's very interesting. I'll move on to Muskani Tahira. She has a question for Akankshi. Muskan, I've asked you to unmute.
Good afternoon, good morning to everyone. I was having a question like, are all the mechanical processes similar in animals belonging to different species, but having the same number of semicircular canals?
When you say mechanical processes, like you mean the mechanism? Do you mean like the mechanism by which that I showed you, is it similar across species? Okay, that's fine. I'll just interpret that that question. So
yeah, she's saying yes.
Yeah. Okay. Yeah. So that's a very good question. Like, is it conserved across species that have three semicircular canals? So we do see. So there's like a really old paper in mouse where they look at hyaluronan synthesis enzyme expression pattern over time, and they also see expression of hyaluronan synthesis enzyme in these. They don't, mouse doesn't have buds. It has the structure called outpocketing, which is similar to a bud, but it's, I think the stiffness and this is going back to the concept that Abhijit described very well. Just the properties of the tissue are different. So they don't have a cavitated structure. They have a luminized structure, and they form these outpocketings instead of buds. And in these outpocketings, you also see hyaluronan expression. Again, in frog, it's similar. They have hyaluronan expression. And in fact, the original paper where they brought out the idea that it could be hyaluronan driven is from frogs. So we think it's likely very, very conserved. But there might be some fine tuning of the matrix properties depending on the size of the aortic epithelium that you need to deform, let's say, to form the canals at the end. But that's a very important question. And the good thing is, like in fish, we can observe these things, and through genetics, we can go back and see in mouse or humans if there is data available that if this gene is mutated, do you have disorders that cause inner ear defects? But we can't do more than that in mouse, and for sure not in humans yet, because the inner ear is not accessible for live studies. You can only go and check mutants from fixed samples to say that, oh, they have some defects. Yeah, that's an important question.
But following to that, Akankshi, I have a question. It's just a thought. It's like, I have no doubt zebrafish is an excellent in vivo quantitative model system. But have you considered using stem cell derived inner ear organoids, where you can also see the aortic vesicles and you can do the structures?
Yeah, stem cell derived aortic vesicle. I've been asked that question. I've been asked that question a lot recently whenever I've presented. So we've tried to, from zebrafish, a colleague of mine tried to culture out the ears. It's not possible. From mouse, you can culture out the placodes, but they don't survive the placode is the embryonic structure that gives rise to the adult sensory organ. So they've tried to culture the placodes, and that hasn't been very successful because it can take a few weeks and it's very hard to keep them healthy. And we don't know yet all the external stimuli you need to keep the organ happy. To directly answer, I'm beating around the bush before I answer your actual question of like, can you do that with stem cell? I don't know. Like, I know like you can make these brain organoids now from stem cells. You can first give them signals that help them become ectoderm. So that's the first step. Then you help them neurulate. So you don't directly put like structures that signals that would give rise to brain immediately. Ear is a sensory ectoderm derived organ, which means that it's not a neural structure, but it's not a classic ectodermal structure either. So it's receiving signals from both. I don't know how successful that would be in terms of culture, but it's completely open to trying. Like if you have if you have some idea of what the signals should be from the brain and what the signals should be from the rest of the tissue, can we make a placode like structure, or can we take a placode from a mouse and try to culture it in a way that it forms the ear? But that's an open question. I would still argue for the community, just because we have young scientists here, that I'm a strong proponent of in vivo research. Yes, it's not a simple system where I can break down to say that these are the minimal components I need to form an inner ear or semicircular canals. But with fish, we're pretty close with CRISPR-Cas9 genetics and all the physical perturbations I can do with the inner ear. We can simplify a system to a great extent and be much closer to what's happening in vivo.
Right. No, I think from embryonic stem cells, we can get the inner ear organized. There are a couple of protocols that we can develop, but just a thought. I totally agree with you.
No, yeah, you can get just the cyst or the luminized epithelium. That's very easy to get from epithelial structures. Like it's similar to the gut, the gut organoids, but to get a functional tubes or canals at the end, I don't know of any studies. Yeah.
So Abhijit, we have got a question from YouTube, because we are also streaming on YouTube. So this is a question by Manish Malhotra. He has a very generalized question on, like, please define traction mechanism and cell migration is due to cell signaling, and how cell signaling plays a role in morphogenesis.
Can you please repeat that question?
He's saying, please define traction mechanism, and if cell migration is due to cell signaling, how cell signaling plays a role in morphogenesis.
Okay, I think it is a kind of a quite long and very, I think if I start from the end, that how cell signaling plays a role in the morphogenesis, I think like the Akankshi has given a fantastic, example of that, right? How the signaling plays a role in the morphogenesis. Also how the migration plays a role in morphogenesis, both the video that I showed during my presentation, as well as multiple images and videos that shown by Akankshi. I think those were the pretty kind of good examples that to create any kind of structure, it is not that the cells are automatically there. Cells were somewhere, some other place. They came together, they moved around, then they formed that structure. So the migration is important there in every aspect of the morphogenesis. And the traction, the first question was the traction. So essentially all these adherent cells, they adhere with their surroundings using their adhesion point. And as Akankshi also said, that they use the actomyosin, so actin fibers, and then the myosin motors to pull in, and that is the cellular traction. And they use that for their movement to know what kind of stiffness, around, to create buckling and all such kind of stuff.
Right. So thank you, Abhijit, for that. I think with that, we don't see any more questions here, but if anyone wants
I
to...
have a quick question for Akankshi,
sure.
just out of curiosity.
And also Shoradeep has his hand raised.
Yeah, he has
Sorry
a raised
to cut
hand,
you.
so we actually do the career-related questions as well, so Shoradeep will take care.
Okay, sure. Sorry, Abhijit, I didn't mean to cut you.
No. So I mean, not directly from your presentation, but just out of curiosity. So you are talking about the different species, they have different number of canals, right? Some 3, some. So what is the evolutionary, the reason for that? Or?
Yeah, I don't know. Like it's very curious. Like lamprey and hagfish, I think they have like 1 or 2. And just to add to that sort of imagination, in sloths, so they have 3. They're mammals. They have 3 canals. The organ is becoming vestigial. Actually, I find it like really funny because they don't move very much, so they don't need to balance all that much. So the evolution process, the evolutionary process is like in the works that it's actually becoming vestigial in sloths. I mean, it's like all evolutionary questions, right? We can speculate a lot in terms of how the structure came into being. So squids don't, as far as I know, squids don't have semicircular canals, so they're invertebrates. But they have these statocysts, which are analogous to our otoliths. So these are the structures that sense gravity. So in my mind, this evolutionary question is that the first thing that both marine and land organisms had to deal with when we started becoming upright and coming away from the ground, or in the case of marine for swimming, was gravity. And then came the evolution of sensing acceleration and direction. So like when we turn, we need this motion. In terms of 1 versus 2 versus 3, I think this is an axis problem. So you this degree, and that degree. I don't know, like there you can think about this problem from so many different ways, right? As an evolutionary biologist, as someone designing GPS for satellites, or mathematician, like how do you think about direction? So I can keep going on and on in terms of how to think about this problem, and I think these will all be speculations. But one of the projects that I would really like to do in my lab is basically try to correlate form and function. And here you can imagine is just the semicircular canal is just the best model system for that. So let's say if you do have problems, and so these 3 canals are mutually orthogonal. So how important or how robust do these angles need to be? How much error can they handle?
That was maybe going to be my next question.
Yeah. That will tangentially answer, like evolutionarily, like do you need robustness in these angles, or can the system cope with a lot of noise? And if I have mutants that result in the formation of 2 versus 3 canals, how is the fish swimming? So you can do behavioral analysis on that. So there's a mutant where the fish swims in circles. Actually, it has defective canal formation. So these are all tractable questions in terms of mechanics, but in terms of evolution, I think of it as more philosophical right now rather than how to address it directly.
Thank you.
Sure.
So I'll move on to Shoradeep. He has a question. So Shoradeep, I've asked you to unmute.
Hi, Akankshi. Great talk, fascinating science. I couldn't help but notice that you had a very unique academic journey. You did your master's in India, your PhD in France, and you're doing postdoc in US. So not only 3 countries, you essentially work in 3 continents. So I was just wondering if you could highlight how different your working experience has been of doing science in 3 different continents.
Yeah, it's been exciting. I'm actually now looking forward to not moving after this move, but it comes with the cost of, like, you have to uproot. So in terms of environment, it's really nice. It builds a lot of moving places, builds a lot of cultural tolerance and appreciation for diversity in a way that is hard to get in one place. In France, my experience, which is, I guess, broadly true for a lot of European labs, is that it's very team-based. So you have an institute where the resources are shared. You have in a way fixed term or limited time in which you have to finish your PhD. So you can't stray too much away from let's say the projects you propose in your first year. So it gives you a sense of like security. I think that you have to finish within a limited time period, but you also end up doing sometimes safe projects. So there's less exploration or less time for exploration. So that's a big difference that is in European lab versus Indian or American system, where time is not the biggest constraint. So this is also then scaled with pay structure, right? Like in India, how the salaries are, or in US, how the salaries are. This is reflective in that's usually not because that's not the limiting factor. People can have longer exploratory projects which have advantages and disadvantages. So I would say that was the highlighting difference for me from a European lab versus India and US clubbed together. I think India and US have a lot of similarities, but yes, in US, it depends on where you are. Like, of course, like I'm in Harvard, there's no dearth of resources, right? So you can be in a smaller university and there is dearth of resources. And again, I was in NCBS in India where there are plenty of resources. So I don't want to compare too much in terms of that the resources are better at one places versus the other, because it depends where you are in US versus India. But I have a strong appreciation for the European system in terms of the respect for time and the cost that is involved with a student or a trainee in general, even postdocs, because that helps you design projects that are likely to be, one, completed, and two, like moving the field a step forward. And with bioRxiv now, I think like we're normalizing the system a little bit as well, where even like if you're not able to finish your PhD in a certain amount of time, if you have that pressure, you can still put something together and upload. So I think we're normalizing the differences between these countries and continents with more open access research. That's like, yeah, I think I went all over the place. There's no easy answer to that question, because even within these continents, it's very place dependent. Like I could have been in different universities with a completely different experience.
Awesome. Thank you. Thank you so much, Akankshi.
Sure. Thanks for the question. Yeah.
Yeah. If anyone else has any question or wants to interact, please raise your hand and I can ask you on mute. Now, with that, I have a question for Abhijit. Abhijit, you have been trained as an engineer in the beginning, and now you're a biologist. How was that transition, and how did you gain the interest and move on to biology? Although you can still use physics and biology now together in your lab.
I don't know if Neha is around or Neha has left, because Neha was, very much there. No, I think she has left.
She just left, yeah.
So Neha was very much there when I joined Jyotsna's lab, right? And as I often say, that I still have that NCBS first, that the small kind of notebook that they used to give to doodle. And when I joined Jyotsna's lab as a postdoc, I still have that one. The first thing written, what is cell cycle? What are the different phases of cell cycle, right? What is actomyosin contractility? What is migration? So, starting from there and to come here. So everything I had to kind of go through, read. And as I was kind of looking for Neha, I mean, all of the people in, so the first of all, I would say a great, deal goes to Jyotsna, that not many people will take someone from a very different background as a postdoc into that lab, right? I mean, initially I did not have anything to contribute in Jyotsna's lab. I was in a completely learning phase, and learning, even like, you know, maybe a intern knows more than me, that kind of learning. So that was one thing. But other than the knowledge base, another example that I keep on giving is that the language, the language. Say, for example, the lab mates, they look under the, you know, microscope and say, oh, your cells are not looking happy. And coming from an engineering background, that was completely, you know, I could not accept that. What do you mean by cells are not looking happy? Give me some kind of number and I can kind of, you know, make it. Then they used to call that, used to say, this antibody is not working in my hand. What do you mean by antibody is not working in your hand? Either it will work or it will not work, right? So it took a lot of the time for me to appreciate that non-linearity of the, you know, the biological system, the ambiguity in the biological system. And also, I think if I think of from the lab mate point of view, say once Jyotsna told me that, Abhijit, you are talking about stress and strain. For me, both things are the same. I am stressed. I am under a lot of strain. They are the same thing. So, and then she used to say that, oh, for me, X and Y are 2 chromosomes, and for you, X and Y are 2 axes. So the language, jargons, everything, the thought process was also so different. So yeah, it took a little bit of time. It is, I mean, I will not say it is an easy journey, but I must say it is an, I mean, incredibly enjoyable journey. That, you know, when you start to kind of link between various different things, maths, mechanics, physics, with this incredible world of biology, it is a hugely satisfying experience.
It is fantastic to hear this. I think that will motivate many of us here who are from a different discipline, want to move to a different discipline, not only to biology. You can be a biologist moving to a physics background.
And like we have many examples, right? With my hugely successful people. So, I mean, it is not that me, you know, who is doing this journey for the first time, right? There are many role models. But I mean, as I said, that it is not easy, but it is immensely satisfying once you kind of, you know, it is almost like you are developing it by yourself. So it is very satisfying.
No, absolutely. I totally agree with that. I think with that, I'll not keep you waiting, both of our speakers, for that fantastic talk and sharing your journey into the scientific research. Thanks a lot, Dr. Akankshi Munjal, Dr. Abhijit Majumder, for accepting our invitation to speak in this platform. And then it's amazing to see so many people joining in on a Sunday evening in India and also early morning in the US. So big thanks to everyone for tuning in. We truly appreciate your support and participation. And please join us.
Thanks for the opportunity. Yeah, I just wanted to add to Abhijit that the future is quantitative biology. So for everyone here, don't worry about what's your background. Yeah, that is the future. Yeah.
No, that's good.
Yeah, thank you, Shaunak. And you know, have you noticed that both of your speakers AM today?
Yes, I did. I did.
Okay, thank you so much. Thank you, everyone.
There are a lot of similarities in the institutions you have worked.
Yeah.
Both from NCBS, Harvard Medical School.
Yes.
It's great to see. I don't know whether you know each other from before.
I've heard of your name, but yeah,
same
never.
Here.
Yeah.
That's great. I mean, that's what we want to promote in the body. Yeah. Well, no, thanks again for doing it. Thanks a lot. Okay. Goodbye.